Hey everybody, how are you doing? Today we are talking about where is diversity located. This is the first of the two where is diversity located lectures, and this one is all about genetic diversity. So in this one we're going to talk a little bit about genotypes, karyotypes, phenotypes, all these slightly jargony words, but by the end of it you should be able to have a decent handle on it. So I'll just start sharing the screen. Let's see which one we do. This one, just share. Okay, so you now should be looking at the lecture. I'll just put the right pointer options. There we go. So by the end of this class, you guys should be able to describe 3 ways to measure genetic diversity. You should be able to apply knowledge of these measures to real situations to measure or compare diversity. You should be able to explain where genetic diversity occurs in organisms. Right, why is that not clicking forward? Okay, so first up, what is genetic diversity? Well, genetic diversity is the number of genetic characteristics within a single species. So often when we talk about diversity, we might talk about diversity of species. How many different species live in an area, for example? But when we're doing genetic diversity, we're looking at the number of different characteristics that exist within a single species. So here's some of the examples. All of these peppers, for example, are the same species. So they're all the same species, but you can see there is a huge range of different what we call traits within this species. Some are yellow, some are green, some are red, some are long and thin, others are short and squat. This is a range of different characteristics which are determined by the genetics of each one of these individuals. Now, when it comes to different genetic characteristics from the species, there's one species that has an enormous range of genetic characteristics and an associated very large difference in physical characteristics, and that is dogs. So all dogs that you know of, everything from a Chihuahua to a Alsatian and boxers, labs, everything in between, all dogs are members of the exact same species. But the genetics of dogs have been heavily manipulated by people through selective breeding over the years. And so we can now produce this huge range of animals with different physical characteristics, different behavioral characteristics, but they're all the same species. And it's because within the dog species, there's an awful lot of genetic diversity present in that one species. And this genetic diversity is differences in the DNA, the genes, the chromosomes of this particular species. So when we talk about genetic diversity, we're talking about the huge range of different genes and different genotypes that exist within a species. And what do we mean by a species? In this case, a species is defined as a collection of individuals that can reproduce with each other to produce fertile offspring. And we'll talk a bit more about this later on, but the basic definition of a species is a group of individuals where they can breed and the offspring they produce are also capable of breeding. And we'll talk about why that's important later in the class. So we might ask ourselves, why is genetic diversity actually important? Do we really need a huge range of different genes in our population? Well, yes, very much so. Genetic diversity is very important because it allows an organism the ability to survive and adapt to change. It means that because you've got these different genes in the population, if, for example, a new disease turns up, there's so much genetic variation within inside the population that some individuals by chance might have a genetic immunity to that new disease and so are able to survive and go on forwards. The most extreme example of this is the Irish potato famine. So the Irish potato famine occurred in Ireland between the years 1845 to 1852. And what happened was, so the Irish loved a potato, and they still do, to be honest, but fair enough, potatoes are great. But the potato was the staple crop of Ireland. It was pretty much what everybody lived off. And this new disease arrived called blight. And potato blight ended up infecting and killing 75% of the total potato crop of Ireland. All of these potatoes that were grown in Ireland were basically one single variety. So they were genetically very similar to each other indeed. That meant that when this new disease, blight, appeared, it was able to rapidly infect and kill an awful lot of the potato crop. So because all of these species, all of the individuals in this species were very genetically similar to each other, they were very unable to overcome disease. Another example that's happening in the modern world is actually the Florida panther. So the Florida panther, due to development in Florida, roads, residential areas, all this sort of stuff, Florida panthers have become spectacularly inbred. They're genetically all very, very similar to each other. The problem with that is they're getting what we call over time, the quality of the genetics goes down an awful lot. I'm trying to put this as politely as possible. And that means that every individual has very, very low survival and very low fitness. They're very susceptible to disease. And also they're not particularly smart. So because they've become severely inbred, the population is in real trouble. What we're going to do now is think about the organization of genetics. And what we're going to do for that is we're going to try and build a concept map. So you get concept maps of this sort of thought exercise, and they get in two basic forms. You have a linear concept map, which was shown here. So basically the idea is, well, you start small, so sangrains all combine together to form a dune. Dunes combine together to form a beach, and beaches combine together to form a coastline. So you can see how we have this linear concept map starting from small up to big. You can also get nested concept maps where it's not in such a linear route, but you do get the same sort of idea. So here's one where we have trees. Trees give oxygen and they give wood. Oxygen is important to humans, it's important to plants, it's important to animals. Wood is used to build houses, paper, and furniture. So this is a nested map, which doesn't have this continuous linear line. But they're just two basic ideas of concept maps. What we're going to try and do now is actually make a concept map for genetics and essentially molecular biology. So I want you to just take a minute, or take 2 minutes on your own, and we'll make a concept map where we're looking at, we've got carbon, cell, chromosome, gene, nucleotide, nucleus, organ, organism, and tissue. What I want you to do is arrange these different factors into starting at the smallest, going to the largest, okay? And I'm just going to time it on my watch. I'll just give you a minute to do that. So remember, just put them in smallest to largest. Okay, just another 20 seconds. Ten, 5, 4, 3, 2, 1. Righty, let's see how we did. So when you try and make your concept map, you should have something that looks a bit like this. So we've got a linear concept map. Start at the smallest. So smallest is carbon. Carbon is an atom. It is basically the very smallest building block of life until you go to the subatomic level, which we're absolutely not going to do. So carbon atom is the smallest. Carbon atoms combine to form nucleotides. Okay, so nucleotides, this ring here contains carbon atoms. Nucleotides, the very, very tiniest building blocks of DNA. So you might have heard about ATCD, so the different types of nucleotides that you can get. Nucleotides combine together to form a gene. So for example, on this piece of DNA, this section here forms one gene. each one of these little bars is 2 nucleotides stuck together, like these two here. Okay. Genes, so the entire strand of DNA combines together to form a chromosome. The chromosomes are all combined together inside the nucleus. The nucleus is contained inside a cell. Cells combine together to form tissues, in this case muscle. Tissues combine together to form an organ. So, for example, let's look at the brain. Brain is pretty much consisted of one tissue, but there are multiple parts. Liver contains one tissue. Lungs contain multiple different types of tissue. They contain the actual lung tissue themselves, and they contain the arteries and the pulmonary bits and all that sort of fun stuff. And then organs combine together to form organisms. In this case, a platypus, probably the most ridiculous organism on the planet. Okay, so this is our concept map of how genetic diversity, and even in some cases molecular diversity, fits in the grander scheme of things. So we go from carbon at the molecular level all the way up to the cellular level, and then all the way up to the organismal level. So this is what we call the levels of complexity of biological organization. Okay, so this is where our bits of sort of genetics sit in. So let's have a think about some of the terminology. So, well, the first bit of terminology I want you to get your head around is a karyotype. So here we have all of our different chromosomes, or a whole bunch of different chromosomes. And a karyotype is the number and appearance of the chromosomes inside a single organism. Can anyone tell me what, or have a guess at what this karyotype is? Look at the number of pairs of chromosomes and tell me what this karyotype is. Or think about it, you can't obviously tell me, but. So this is the karyotype for a human female. We know that because it has 23 pairs of chromosomes, like humans do. And then we know this one is a female because it has 2 X chromosomes. So males in humans have, and most mammals, have XY. So that if it was a male, this chromosome would be sitting over on the Y. But it's a female because it's got 2 X chromosomes. So this is the basic karyotype of a human. Every single one of us has 23 pairs of chromosomes, hence the name 23andMe, the genetic company that will tell you your genes. It's based on the idea that you, me, we have 23 pairs of chromosomes. Those 23 pairs of chromosomes determine who we are. So this is a karyotype. Now, within the karyotype, you can get chromosomal alterations, and this can lead to some changes in physical characteristics. So the chromosomal alterations you can get are things like change in the size of different chromosomes, change in the shape of particular chromosomes, change in the banding of chromosomes, and changes in the number. So, for example, these two karyotypes here show 2 karyotypes that are associated with different syndromes. So this one, if you can see on this X chromosome here, there's a little tiny almost notch on the end of the X chromosome. This causes a syndrome called Fragile X syndrome. It usually affects males because it only tends to happen on 1 chromosome. And because females have 2 X chromosomes, then if the other X chromosome does not show this Fragile X, then there's no physical change at all. But Fragile X syndrome is associated with a sort of long forehead, longer ears, and it is associated with some form of developmental impairment. This karyotype change on the bottom is one that a lot of people will actually be familiar with. This is a duplicate in pair 21, and that leads to Down syndrome. So if you have an extra chromosome, you tend to get Down syndrome, which has the characteristic a mild physical and developmental impairment. I've realized I've made a mistake on this one because this is XY and it's a female that I've put a picture of. So that should be 2 X's, sorry, or I should have put a male picture there. But Down syndrome is associated with the characteristic change in the face. very mild physical and developmental impairment. But in both cases, both Fragile X and Down syndrome, these people will survive quite happily. They are functional members of society. So even these small changes in chromosomes, yes, it does lead to some characteristics, but it's in no way fatal. Okay, so you can have these changes in karyotypes, and what happens? Righty, we'll just do a fake clicker question for a second. So I want you to think about this. So what is the relationship between an organism's complexity and the number of chromosomes? Just think through this. So A, more recent species, such as humans, have more chromosomes. B, all species have the same number of chromosomes. C, there is no clear relationship between the age of a species and its number of chromosomes. Or D, older species have more chromosomes. So we'll just think about this for another 15 seconds and see what you think the answer is. Okay, 3, 2, 1, go. Well, the actual answer is there is no clear relationship between the age of a species and its number of chromosomes. And some of these examples are pretty stark. So let's have a look at these two karyotypes here. So this karyotype here, what do we think this is? There's a bit of a clue at the top. This is a human male. We've got the same 23 pairs of chromosomes. We've got the X and we've got the Y. So there we have a human male. In this case, we'll say me. This is going to be, for the purpose of this observation, my chromosomes, my karyotype. Now let's have a look at this one here. This is much more complicated. First of all, we've got 78 pairs of chromosomes, and then we've got these 6 little magical things tacked on the end. This looks like a very complex karyotype indeed. So what species would we think this is? Well, we think it might be something incredibly complex. It might be a very large animal. It might be a giraffe or a whale or something like that. In fact, it's actually a common carp. So I would argue I'm slightly more sort of complex, for want of a better word, than a common carp. But my karyotype is much simpler than that of the common carp. So there isn't any real relationship between the position of an organism on the evolutionary tree and how many chromosomes it's got. In addition, even within some closely related groups, the chromosomes are very, very complicated, can be very different indeed. So for example, here we've got 4 different karyotypes. The top 2, we've got 20 pairs of chromosomes and 1 single sex chromosome. The bottom 2, we've only got 6 pairs of chromosomes, and we've got these weird neo-X, neo-Y sex-determinant chromosomes. So you can see we've got very, very different karyotypes. But all of these karyotypes belong to one group of species, and that is funnel web spiders. These are all a different species of funnel web spider. So even though they're very closely related, and let's be honest, are absolute nightmare fuel, their karyotypes are very different from each other. Now, just as a slight aside, I used to live in a part of Sydney called Lane Cove, which has got a very famous Lane Cove Park. Lane Cove Park is, the reason it's famous because it has the highest density of funnel web spiders anywhere in the world. And there's a little bit of an argument about whether or not the Australian funnel web spider is more dangerous than the Brazilian, what's the Brazilian one called? It's just popped out of my head. Oh, is it the wandering spider? Yes, the Brazilian wandering spider, if that's more dangerous than the funnel web spider. They're both nasty things, but to be honest, even though they are among the most dangerous of venomous spiders in the world, no one's been killed by one in Australia for 10 years. They're pretty fine. They were actually running out of anti-venom when we lived there. So they were even telling people, if you encounter a funnel web spider in your house, please try and catch it with a piece of Tupperware because they need it to produce the anti-venom. So they were asking members of the public to go out and catch these things, not very dangerous. If you do want to have a think about it, everyone seems to think Australia is full of things that will bite and sting and murder you in 1,000 horrible ways. But actually, the animal in Australia, or the wild animal in Australia, that causes the highest amount of deaths is this thing, the Australian magpie. It doesn't have a poisonous bite, it doesn't have a sting, it doesn't even have teeth. But magpies become incredibly territorial in the month of September, and they will attack anything that goes into what they perceive to be their territory. So as a result, cyclists in particular, while cycling along, get swooped and attacked. They will literally bite you on the top of the head, and they will draw blood, attack you, and cyclists then end up veering into the road and getting hit by cars. And this is recorded as a magpie-related mortality. I got attacked by these a few times. If you've got a few minutes on YouTube, it's well worth a bit of your time having a look at swooping magpies attacking cyclists. It's generally quite entertaining. So that was a karyotype. Now we're going to talk about the genotypes. So genotypes are the genetic makeup of a specific organism. And what we can use this for is, genotypes are your specific genes that are contained in your DNA. We can use the genotype to make a genetic fingerprint. This is something that's often used in crime now, and it's how 23andMe works. And we can use the structure of these genetic fingerprints to actually build what we call a phylogram or a family tree. And what a family tree or phylogram does is it looks for how closely related the genetics of different people are or different organisms, and they can actually group them into families. So for example, you end up with a thing that comes out like this. What they generally say is, it's not how different from each other in this direction you are, it's when you split off in this direction. So for example, this is going to be the point, all the organisms we're talking about, all the individuals are basically branched together here. This split might be two different extended families. This might be then one brother and one sister, and then they split, and this is the offspring of those two, and such and such. So it goes in this direction, not differences in this direction. But so this is how we can use our genotypes to build family trees and also identify individuals through genetic fingerprints. Our genotype also affects what we look like. So this is called our phenotype. And our phenotype, so our physical characteristics, are influenced by our genotype. They're not absolutely determined by your genotype. There is some aspect of changes through a personal and an individual organism's life history, but our genotype heavily influences our phenotype. So this effect of genotype, so what our genes contain, on phenotype can lead to some embarrassing questions. I apologize for the age of these photos, but it kind of illustrates it best. So does anybody know who this is? This is Prince William, current husband of Kate Middleton, and heir to the throne. This is Prince Charles. Prince Charles is Prince William's father. Prince Charles fathered Prince William through Lady Diana Spencer. These two, this is his dad, basically. So that's Prince Charles and Prince William. Does anybody know who this is? This is a very old photo indeed. He's aged much better than his brother has, it has to be said. So this is Prince Harry. Now, Prince Harry is Prince William's brother, and Prince Charles is allegedly his father. So Prince Harry was born again by his mother was Diana Spencer. He's now married to Meghan Markle, but Prince Charles is allegedly Prince Harry's father. There is one issue with this, though, and that is that Lady Diana had a very public affair, well, she had multiple public affairs, very public affairs about the time when Prince Harry was supposed to be conceived. And one of the people he had an affair with, or she had an affair with, is this person, Major James Hewitt. So if you were to just look at these two, you can see the eyes, the nose, the shape of the mouth is very, very similar. They share an awful lot of physical characteristics. This guy is meant to be this guy's father, but they don't share any of those physical characteristics. He does appear, however, to share quite a lot of physical characteristics with Major James Hewitt. So there are some questions being asked, and not surprisingly, neither Prince Harry, Prince William, or Prince Charles have volunteered for 23andMe, because I think that would lead to some very embarrassing questions. So after that little bit, let's just do another little fake clicker question. So what best describes the relationship between genotype, phenotype, and the environment? So is it A, genotype is our genes. It exactly determines our phenotype, which is the structure of our chromosomes. Is it B, genotype is our genes. Our genotype interacts with our environment to determine what we look like, i.e. our phenotype. C, the environment we live in will change both our phenotype and our genotype within our lifetimes. Or D, all individuals of a species have the exact same genotype, but environmental changes make them behave differently. Just give you 10 seconds to have a think about which one this is. Okay, so correct answer is B. Genotype is our genes. Our genotype interacts with our environment to determine what we look like, which is our phenotype. Interestingly, this one, letter C, the environment we live in will change both our phenotype and our genotype within our lifetimes. That used to actually be thought of as a valid scientific hypothesis. People actually believed that they didn't know what genes were, but they thought that the characteristics and the mechanism that determines an organism would change within its lifetime. It was known as Lamarckism, but that's a bit of an aside. Okay, so what we're going to do now is talk a little bit about genetic versus environmental variation. So remember, a phenotype is an organism's visible traits, and they are also affected by the environment. And this is known as nature versus nurture. Let's think of a basic example of this. So here we have one particular species of plant, okay, and it seems to exist in multiple different sizes. So we see on this plant, it's got a whole bunch of different sizes. It's the same species, but it seems to be a whole bunch of different sizes. And when we look at where these different sized plants exist, we see that we find changes in the phenotype, changes in the organism's visible traits, as we move up and down a mountain. So right at the very peak of the mountain, we've got very, very tiny plants. About sort of 2/3 of the way up, or about 1/3 of the way up the mountain, we have the biggest plants. Right at the very bottom, we have the smallest plants. So we can think about, okay, why are we seeing this pattern? Why are we seeing tiny plants at the top, massive plants in the middle, and smaller but bigger than the top at the bottom? Well, what we can do is we can elucidate whether this is due to nature or nurture, so whether it's due to changes in the genetics of these plants at different places, or if it's due to changes in the environment in which these plants live, by doing what are known as transplant or common garden experiments. So in a transplant experiment, what you do is you take individuals from all these different places. You take an individual that was growing here, or you take the seed from it and you move that seed up to the top, or you move that seed down to the bottom, and then you grow the individual at that location. So that's a transplant experiment. You're transplanting seeds from one place and you're moving to another place and seeing what that plant looks like. Common garden experiments are basically where you take seeds from individuals from every single location. You transplant those seeds to the exact same environment, so like down in a valley somewhere, you grow those plants and you see what they look like. And using these transplant and common garden experiments, we can understand whether these changes in size that we're seeing are due to changes in the plant's genetics, or if they're due to changes in the environment. And so, for example, we can end up with results that look a bit like this. So let's start out in our transplant experiment. So if the variation that we're seeing is all environmental and it's not genetic at all, what would we expect to see? So we're taking seeds from these 3 different locations, bottom, middle, and top. We're transplanting those seeds around, and then we're seeing how the individuals grow. If the changes that we observed in phenotype are all due to the environment that the plant grows in and nothing to do with genetic, what would we see? Well, we'd see something that looks a bit like this. So here we have, this is our lowland garden, And this is our lowland plant, our mid-elevation plant, and our alpine plant, so the plant from the very top. If all of the environment, if all the variation of phenotype we see is due to the environment, we'd expect plants grown in one garden to all look exactly the same, regardless of where the seed was collected from. So a lowland plant grown in a lowland garden should look exactly the same as an alpine plant grown in an alpine garden, because all of these seeds have been taken, they've been put into one environment, and because the environment is determining their phenotype, they should all look the same. And then same for mid-elevation, regardless of where you're collected from, all the plants should look the same height. A lowland plant, mid-elevation, alpine plant should all be the same size. At the alpine garden right at the top, we'd expect them all to be tiny, just like the alpine plants we observed in nature. So that's if it's all environment and not genetic. What about if it's all genetic and there's no influence of the environment on the change of these plants? Well, if this is the case, what we'd expect to see is that the plants collected from the lowland plants would look exactly the same as lowland plants if they're planted in the lowland garden, if they're planted in the mid-elevation garden, or if they're planted in the alpine garden. Same for the mid-elevation plants. They should look exactly the same if they're in the lowland garden, the mid-elevation garden, or the alpine garden. And finally, same for the alpine plants. This is because we would expect to see this because, in this case, differences in the phenotype that we observed are all due to genetics. It doesn't matter where you place a plant, what it looks like is determined by where you collected it from, not by where you placed it, because plants from different locations have different genetics, and so as a result, we'd expect that plant to look the same regardless of where you plant it. But what we often see is it's a mixture of both. We see both genetic and environmental changes. So here, let's look at the lowland plants first. Lowland plants, they grow very well indeed in the lowland garden because their genes are well suited to a lowland environment. They grow okay at mid-elevation, but they grow very, very badly indeed at highland. They're just not designed to live up there. Conversely, the mid-elevation plants, they grow okay in the lowland garden. They grow really well in the mid-elevation because that's where they're designed to grow. That's what design's the wrong word. That's where they have adapted to survive. And they don't do so well at the high alpine. Okay. So we start to see these changes. The interesting one is actually if we look at the alpine ones. So the alpine group, remember these came from the alpine garden, don't do very well at the lowland. In this case, they appear to do very well in the mid-elevation, and they do pretty well in the alpine as well. We might see this sort of pattern if this plant has got good conditions here, but when it tries to grow here, it's outcompeted by other plants. So as a result, it's just not able to grow, not because it's not suited to the conditions, but because there are other plants that are better suited to the conditions, and they end up excluding it. So this is what we'd expect to see, a mixture of genetic and environmental. So remember, if it's all due to the environment, no matter where the plant was collected from, it will always grow exactly the same in different gardens. If it's all due to genetics, that means that it's only low elevation plants always look the same regardless of where you put them. Mid-elevation plants always look the same regardless of where you put them, and same for alpines. If it's a mixture, you tend to see a bit of a mess. Okay, and that's how you can identify whether or not differences in phenotype that you see are due to differences in genetics or differences to the environment. Okay, and so we'll just sort of end on these sorts of ideas. What we're going to talk about now is how changes in genotype can actually produce new phenotypes. So small little changes in your genotypes, what we call small mutations, can produce new phenotypes, new sort of physical characteristics. But when we talk about this, one of the ideas that gets bandied around an awful lot is like, okay, change in genes, that's how you evolve totally new structures. But what actually tends to happen is when we talk about change in genotype, rather than evolve new structures, it tends to just turn on and off developmental genes. So what it means is you have your basic structure, but then as the organism is developing as an embryo, different genes get turned on and off for different amounts of time, and that tells different structures to become different sizes. So for example, this is a whole bunch of the arm and the arm bones or limb bones of mammals. We've got human, horse, cat, bat, bird, and whale. And within these bones, the same colors represent the same bones. What's happened is no new bones have been made, they've just changed in shape. So this is a human. Here's our main arm bone, here's the radius and the ulnar, here's all the wrist bones, and here are your phalanges, your fingers. You can see these same bones exist in the horse, but things like the fingers, most of them have been lost. We now just have one big long finger. The wrist is much higher. The cat, it looks actually most similar to the human out of all of these. The bird, I said mammals earlier on, that's a misnomer because a bird isn't a mammal, but there we are. The bird still has the same bones in it, but the most extreme are things like the bat and the whale. So let's specifically compare something like the whale and the horse. Look how tiny the arm bones and the radius and the ulnar have become, but look how massive the fingers have. This is because this is now the flipper of a whale. Compare that to a horse. We've got this is much longer, this is much longer, and we've got multiple fingers. So it's the same bones, it's just been acted on to change slightly in shape. Then we can ask about, well, how do we know these are the same structures? Well, what we do is we follow these organisms through development, and we can see how they change. So here we have a whole series of developing embryos. What we can see is at the very, very early stage, all of these embryos look very, very similar to each other. But as development proceeds, they differentiate. And so by the time the organism is about ready to be born, all of them look radically different. Snake, chicken, possum, cat, bat, and human. Because what's happening is evolution acts on developmental genes, and it's much easier. It can actually quickly produce new forms with far less risk. And this is a principle called evo-devo, evolutionary developmental biology. And I've put this slide up on line as well. It's well worth having a bit of a giggle at this slightly ridiculous YouTube video where they cover the whole field of evo-devo very, very rapidly to a slightly daft song, if you get time. So to one last quick fake clicker question. So how does evolution generally act on phenotypes to create new, act on genotypes to create new phenotypes? So mutations in genes cause the production of entirely new features. B, there is no relationship between genotype and phenotype. C, the environment will change our genes during our lifetime, meaning we grow new features. Or D, changes in developmental genes change the form of existing features. Just give you 5 seconds on that. So the correct answer is D. Changes in developmental genes will change the form of existing features. Okay. Okay, that's as far as I wanted to get to today. Hope you enjoyed learning a little bit about what genetic diversity actually means, and I will see you next time.